Welcome back. We will now be starting the chapter of the protozoans. So up until now, we've covered the helminths, and that is all test 1 material. From here on out, all the following parasites will be on test 2. So the protozoans are the lowest form of animal life that exists. These are all unicellular organisms, and they are also eukaryotic, meaning they contain at least one nucleus. I've outlined a little bit of what we're going to talk about for the protozoans, just to give you an introduction. So we will cover 7 intestinal amoeba, 2 tissue amoeba, 1 ciliate, and several categories of flagellates. And then what's not listed here are the apicomplexa. So remember I said in the first lecture, the introduction lecture, that amoeba, these are categorized by their motility mostly. So amoeba move by pseudopod, ciliates use their cilia for motility, flagellates of course have a flagella, and then the apicomplexa do not have a means of motility necessarily. So for your amoeba, these are the ones we will talk about. For the intestinal amoeba, Entamoeba histolytica, Entamoeba dispar, Entamoeba hartmanni, Entamoeba coli, Endolimax nana, Iotamoeba bushleii, and Blastocystis ominis. What's interesting to note is not all of these are pathogenic. So why do we need to learn them? Well, because finding them would suggest that the patient has ingested contaminated food or water, which is sort of a surrogate marker to clue the physician in that there might be pathogenic parasites in there as well. And also so we can be able to correctly identify and report the pathogenic versus the non-pathogenic, because they will, as you will see, look very, very, very similar. So general characteristics of our intestinal amoeba. Identification is based on several things, most notably size. So remember I've mentioned that ocular micrometer that is vital to the parasitology lab. The scale that's in the eyepiece of the microscope. You have to be able to measure these trophs and cysts, because that is a lot of times the only thing that's going to differentiate one from another. We're going to be counting nuclei. We're going to be looking at the nuclear structure, the presence of anything in the cytoplasm, any internal structures, and also motility. So that wet prep is also extremely important for those loose, watery stools, because if there's a motile parasite in there, that would be very, very helpful for correct identification. And if you recall, in the wet preps, we mix some of the sample with saline and some with iodine, and iodine will help to stain some of the nuclei. So it's very important that we talk about these intestinal amoeba, because this is the third cause of death due to parasitic infections worldwide. So first is malaria, which we haven't talked about yet. Second is schistosomiasis, caused by what? Remember, caused by our blood flukes, the genus Schistosoma. We had Mansoni, Japonicum, and Hematobium. And then number 3 would be amoebiasis, caused by Entamoeba histolytica. So about 40 to 50 million people are affected, with about 40,000 to 100,000 of those actually dying from this infection. The intestinal amoeba, as the name suggests, live in the large intestines. The life cycle stage is pretty simple, so you can breathe a sigh of relief for a second, because I know those helminth life cycles could be fairly complicated. The amoebas you'll see are very simple with just two life cycle stages, a trophozoite and a cyst. The trophozoite is the motile, feeding, reproductive stage of the parasites, and it reproduces by asexual binary fission. The cyst is the environmentally resistant, infective stage. It's the non-feeding stage. The cysts have protective cell walls, so they can remain viable for long periods of time outside the body. We get infected with intestinal amoebas by direct fecal-oral route of transmission. So this means by consuming contaminated food or water that has those infective cysts. So once we ingest a cyst, the cyst will quote-unquote hatch into the trophozoite form in the large intestines. This is properly termed excistation. The trophozoite will colonize the colon where it feeds on our foodstuffs, and it's able to reproduce fairly rapidly. Now, when the conditions dry out, or there's not enough food or too much food, or not enough oxygen or too much oxygen, or the pH changes, the trophozoite is going to want to transform back into the cyst. So this is called encystation. So we had excistation and encystation. So the cyst can be formed and excreted in the feces. Now, trophs can't survive gastric juices. They're pretty delicate and fragile. So remember, we're more likely to recover trophozoites when the patient has loose, watery stools, constant diarrhea, then we're more likely to see the trophs. But in formed stools, we're going to see the cysts. So here's a great picture. Let's talk through this. At the top, we have human ingestion of infective cyst and contaminated food or water. Excistation will occur in the intestines. It'll transform into the trophozoite, and the trophozoite will feed and reproduce. From here, the trophozoite can be shed, so we can see this diagnostic stage in the sample. It can also be shed and reach the environment in which it would encyst, go back into the cyst form, so it can survive outside the body. And then this could be the way that another human gets infected. Now, specifically for Entamoeba histolytica, okay, only Entamoeba histolytica, we can see this extraintestinal stage in which the parasite can leave the intestines and invade other organs, most notably the liver, and cause hepatic abscesses. So we'll get to that in a second. But in general, you'll see it's just troph and cyst, troph and cyst. It's that simple, just those two life cycle stages. So again, the infective stage of parasite development in amoeba is the cyst. The diagnostic stage is both. So we can see both trophs and cysts in the stool, and we need to know what both of them look like for each parasite. So let's get started with talking about each one individually, starting with the most important one, Entamoeba histolytica. This is found worldwide, the highest prevalence being in the tropics and subtropics, although we do see it here and in developed countries. It is the number one major intestinal amoebic pathogen. It is possible to have an asymptomatic infection with Entamoeba histolytica, but those are the dangerous people because the colonization is occurring and those people are passing cysts, so they can infect people within close quarters, or even it can be sexually transmitted. So it can be as mild as just cramping and tenderness, although the major manifestation of it is called amoebic dysentery, in which the patient has bloody, mucousy diarrhea, very severe, more than 10 stools per day. So I mentioned that extraintestinal phase, and this is unique to Entamoeba histolytica. This organism is capable of leaving the intestinal lumen. It can break down the intestinal wall, get into the bloodstream, and go to other organs. And when it does this, when it eats away at the intestinal mucosa, it forms what looks like a flask-shaped ulcer. So that's a key term here for Entamoeba histolytica. If you see the term flask-shaped ulcer, you know right away what they're referring to. So it can go to other organs or tissues like skin or lungs, but the most common and gravest complication would be that it enters the liver and causes hepatic abscesses. So I mentioned that it can be sexually transmitted. The most common population it's seen in is homosexual males, but truly anyone who practices anal, oral, sexual intercourse can transmit this, because remember, the cysts are infective when passed, and if someone else ingests the infective cyst, that's all it takes. It's infective right away. So trophs and cysts may be passed in the stool, but trophs are not infected. And the cysts are very environmentally resistant, meaning they can live as much as a month in the environment. They do prefer a moist environment, so drying or boiling the water or chemicals will destroy it. But it can absolutely survive in the environment. It's got a thick shell, a thick cell wall. So another picture, again, this might get redundant because it's pretty straightforward. We have a cyst that can get ingested. The cyst is the infective stage, but it can also be the diagnostic stage too. Excistation will occur, so it transforms into the trophozoite, which is the feeding, motile, reproductive stage. So a troph can get passed in the feces, and this is the other diagnostic stage, or it can encyst, transform back into the cyst. And it's showing here, if you see the letter C, that extraintestinal disease, where it can leave the intestines and it can cause abscesses in the liver, but it can also cause lung abscesses and even get into the brain tissue. So what does this guy look like? So starting with the trophozoite form, you don't need to memorize the size, as in the numbers. What you need to know is relatively how big is it or how small is it compared to other parasites. Okay, so keep that in mind. The trophozoite, remember, is the motile stage. So this, if you were lucky enough to see this parasite moving in a wet prep, it exhibits directional progressive motility via pseudopods. What that means is it looks like it's on a mission. It's not just randomly moving around and going in all kinds of directions. It looks like it's going in one direction in particular. It's got one nucleus, and it's described as a bullseye nucleus. So there's a central karyosome surrounded by evenly distributed peripheral chromatin. A karyosome is a small mass of chromatin made of DNA material within the nucleus. And then so in addition to that chromatin material, you also have peripheral chromatin around the surface of the nucleus. And it looks like a little perfect circle. The next bullet is very important. This trophozoite of Entamoeba histolytica may contain ingested RBCs. This would be diagnostic if you were to see this on a sample. No other parasite is going to have ingested RBCs. And this term is erythrophagocytosis, meaning it's phagocytizing RBCs. And it would make sense that it would be in the trophozoite because that's the feeding form. So you would not see ingested RBCs in the cyst. You would only see it in the troph, and it's unique to Entamoeba histolytica. Okay, you need to spend some time with these black and whites because it's showing what a perfect picture of each parasite looks like. In real life, it is very hard to see these things. So if you don't even know what the black and whites are, you'll never know what they are on a real slide. So here's a finger-like pseudopod extension. You may or may not see this in the troph. Okay, trophs can be round, but sometimes they'll have these extensions, meaning we made the wet prep. It got fixed during its extension of the cytoplasm, which is pretty cool. This is your bullseye nucleus. So there's only one nucleus in the troph. It's got that central karyosome. It's right smack dab in the middle, and it's surrounded by evenly distributed peripheral chromatin. Now, the cytoplasm may contain vacuoles or debris or bacteria because, again, this is the feeding stage. So it's possible that the cytoplasm looks kind of dirty and messy. So in real life, this is what a stain of it looks like. You are able to see some extensions, a pseudopod. There's your centrally located karyosome and evenly distributed peripheral chromatin. Here it is again. And here we are again. This is still a troph because there's one nucleus. So just because it's rounder in shape doesn't mean it's a cyst. Okay, again, a rounder shaped troph, that's fine. This is an ingested RBC. This is erythrophagocytosis. This would be diagnostic. If you saw this on the slide, you would be able to call this Entamoeba histolytica. That in combination with that perfect bullseye nucleus. Here it is again, evenly distributed peripheral chromatin and a centrally located karyosome. I always tell students this is sort of a trade-off because with the helminths, the life cycles were fairly complicated, but those eggs and those larvae, those proglottids, the scolices, all that is very easy to see. You don't have to look too hard to diagnose helminths. The protozoans, on the other hand, are very difficult to see, but they have the simpler life cycle, so there's sort of a trade-off there. So that was the troph. Let's talk about what the cyst looks like. The cyst is a little bit smaller. It can have up to 4 nuclei, so 1 to 4 nuclei in the cyst. Although if you see something with just 1 nucleus, we're going to call it a troph. If it's got 2, 3, or 4, then it's definitely a cyst. The nuclear characteristics are the same as the troph, so we still have the bullseye nucleus, a central karyosome and evenly distributed peripheral chromatin. What can also be in the cyst are called chromatoidal bars, which these are made up of RNA, and they are cigar-shaped, meaning the ends are nice and rounded. They're nice, smooth, rounded chromatoidal bars. So here's your black and white, your perfect picture of what it would look like. Bullseye nuclei, it can have up to 4, and your rounded ends of the chromatoidal bars. That's not a great picture because we only see 1 nucleus. But here you can see 3, and there's a very prominent chromatoidal bar in there. You really have to use fine adjustment to make sure you're counting all your nuclei. Quite often, you'll look at something and it looks like there's only 1 nucleus or maybe 2, and then you'll do slight fine tuning and then 2 more nuclei will pop out. So you've got to make sure you're really looking at each organism very carefully. So that is what the cysts and the trophs look like. Now we can also do serological diagnosis for Entamoeba histolytica. This is most helpful in the patients with extraintestinal disease. So they have liver or lung or other involvement. So in 90% of those patients will get a positive antibody test. However, you cannot distinguish between an old or a new infection, just like with other antibodies for syphilis. You don't know if it's past or present. We can also do antigen testing on the stool. So we can look for antigens that are specific for this parasite, surface proteins or adhesion lectins on the parasite. This is the most definitive way to diagnose Entamoeba histolytica. It is the only way to distinguish it from Entamoeba dispar, which is the next parasite we'll talk about. And you can also do molecular testing, actually look for the specific DNA sequence. This would tell you, antigen testing would tell you that there's absolutely a current infection. Entamoeba dispar is a very short discussion. It is morphologically identical to Entamoeba histolytica. So it has a bullseye nucleus with that central karyosome and even peripheral chromatin. It is the same size. However, it is non-pathogenic, meaning the patient isn't going to have amoebic dysentery. They're not going to have an extraintestinal disease from it. They're not even going to have abdominal pain or cramps or anything. It's non-pathogenic. It does not ingest RBCs. So remember, no other parasite does this except Entamoeba histolytica. So you will not see ingested RBCs. Like I said, it's non-invasive and it's serologically different. So the surface antigens are different. We can distinguish them that way. The motility is also non-directional. It's aimless. It's not progressive motility like Entamoeba histolytica, although sometimes you don't get to see motility, and not Entamoeba histolytica isn't always going to ingest RBCs. So quite often when we see something that looks like Entamoeba histolytica, we have to report it E. histolytica / dispar because there's no way to tell just by the slide. They are morphologically identical. So they have to order more tests to decide which one it is. Then very quickly we can cover Entamoeba hartmanni because it is also looks the same as histolytica except it's smaller. The size is significantly smaller. So it's called small race Entamoeba histolytica. It is also non-pathogenic and the motility is non-directional. So once you get a handle on Entamoeba histolytica, you actually can easily describe Entamoeba dispar and Entamoeba hartmanni. So this also has the bullseye nucleus with the central karyosome, even peripheral chromatin. The only difference here is size, and it is also non-pathogenic. And of course, we will not see erythrophagocytosis.